So welcome everyone today. Thank you for coming out on this rainy day. I know some folks are getting ready to watch the Super Bowl. Thank you for coming and joining the Passion for Native Plants instead. So thank you for that. We have a few presenters with us today. We have Leigha Henson, who's here in person, who'll be presenting. Then we have Erin Eichenberger, Lydia Cox, and Lauren Wilhite, who will be presenting virtually through Zoom. So bear with me as we get all of their different presentations up on the screen one after the other. If I haven't met you before and I see some new faces, my name is Craig Maxwell. I'm the local chapter chair for the Southern Piedmont chapter of North Carolina Native Plant Society. Our goal as the NSCU Native Plant Society is to promote the enjoyment and conservation of North Carolina's native plants through education, protection, advocacy, and propagation. We provide scholarships to university students, which we'll be talking about today with the Shinn grant. We do educational chapter meetings here once a month, typically the second Sunday of every month. And we do field trips. We'll get talking about some garden tours and some hikes we have planned this spring, and hopefully you all can make it out to that. If you're interested in becoming a member, ncwildflower.org is how you can, if you can take a look and see what sort of membership you're looking for, whether it's individual from $25 up to, we have 3-year or lifelong memberships for people if you'd like to make that sledge. With no further ado, I'm going to go ahead and introduce Leigha Henson, who'll be our first presenter today. So brief background on the Shinn grant program. Deborah Murray, who's with us, helps organize that. We have a few grants and a few funds that we have on our website. The Zawatsky Fund was the other one that we were promoting at the end of last year. That's more of a land grant. But the Shinn grant fund goes to sponsoring any research being done in North Carolina on native plants. So we had, I'm not sure how many we had last year that we gave out, but there's scholarships of up to $1,000 to help with research. So we have 4 of those recipients. Some of these are 2022 recipients, some are 2023 recipients, and they're here to talk about the work that they've been doing. So we'll kick it off with Leigha Henson, who's from Appalachian State. And then on Zoom, we have Erin Eichenberger from NC State, Lydia Cox from Duke, and then Lauren Wilhite from NC State as well. So I will go ahead and turn it over to Leigha. Thank you.
Okay, so yeah, today I'm going to talk about my thesis research, which is the warming effects of several common moss species in the Southern Appalachian Mountains. And again, I am a master's student at AppState University, and my advisor is Dr. Howard Neufeld. So to kind of give a little bit of background, the Southern Appalachian Mountains is a biodiversity hotspot, especially for bryophytes, with about 386 species of mosses just in this region. And mosses play a lot of ecologically important roles, such as preventing erosion, retaining soil moisture and nutrients, serving as habitat for fauna, and are often pioneers in ecological succession. They can be found in a wide variety of habitats, such as deep shade, like those on the left. The 2 on the left here are deep shade, kind of understory species that are found on down logs or rocks in the forest. And these are my 4 study species on the bottom. And then on the top 2 on the right are open habitat sites where I see those mosses on the bottom right. So Ceratodon purpureus forms dense clumps, really small stems in really dense clumps to conserve moisture. And then Polytrichum juniperrinum, kind of a bigger version of Ceratodon, so just forming bigger clumps. So mosses actually don't have stomata, which vascular plants do. So the top photo is a vascular leaf surface with stomata. And in the bottom picture is Ceratodon purpureus, just a leaf in a microscope. It's only 1 cell thick. So you can see each of those cells and the chloroplast in the cells, which is really cool. But they don't have stomata. So that water is directly absorbed through their semi-permeable cuticle on their epidermal cells. And they're poikilohydric and ombrotrophic, which are some really fun words. So basically that just means that they will quickly desiccate and dry out, and they only remain metabolically active under sufficient moisture contents. So this is a video, I'll show that in a second, but desiccation tolerant mosses, or those found in open habitats, They can utilize different leaf arrangements to conserve moisture, as well as some species actually have overlapping leaves that form little water holding pockets, which is really cool. So this is Polytrichum juniperinum, and I want to show you a video of it drying. So this is a time lapse. It's only about 30 seconds long, but it's a 40-minute window, so you can actually see it dry in real time, which is really cool. Wow. So it forms, so their leaves just go straight against the stem, just parallel to the stem. So it reduces that surface area and it conserves a lot of moisture that way. And then at the end, I'll spray it again and you can see how fast it opens up.
That took place over 40 minutes.
40 minutes, that's it.
Did you have a convection oven going?
Wow. No, this is just, yeah, it's just so silly. This is just in the lab. Pretty crazy. No, this is just, so we've discovered that there have been no studies on moss ecophysiology in the Southern Appalachian Mountains. And this is really important because understanding their responses to different changes in moisture content and light levels could be really crucial to predicting how they might respond to climate change. Okay, so excess water on the leaf surface, since that water is directly absorbed into the epidermal cells, excess water can actually prevent CO2 from diffusing into the leaf, and it can actually inhibit photosynthesis a bit. So I just kind of made a little diagram. That's a Thuidium delicatulum leaf. So that's one of those understory mosses under a microscope. So that this top picture has a really thick layer of water, like a thick film of water on its surface. So that's a fully saturated moss. And you can see that CO2 doesn't as easily absorb directly into the leaf. Whereas on the bottom picture, you can see that as the moss is allowed to dry out a little bit to an intermediate water content, more CO2 can kind of diffuse into the leaf and photosynthesis will peak. And then as it dries further, it'll just decline. So I measured mosses drying over time, basically just let them dry in the lab and weighed them every so often to kind of see how much water they were losing with over time. So the x-axis here is time, and then relative water content, just basically how wet they are. 100% relative water content is a fully saturated moss, 0 is dry. And you can see that they dry pretty fast. Within about 40 minutes, they're really not changing in weight anymore. But you can see Ceratodon purpureus, the bottom graph there, takes almost 2 hours to fully dry. And that could be because of its dense clump formation. So it's conserving a lot of moisture, which is really exciting. And then Polytrichum juniperinum, that second from the bottom, the yellow graph. So that one you can kind of see it stops losing moisture at around 30% relative water content. And again, that might be because of the leaves reorienting to conserve water. So it's kind of adapting to different stress levels. So this is how I measured photosynthesis of mosses, which was pretty complicated, but really fun to learn. So these are each of the 4 species in the cuvette, and then I used an LI 6800 from Leigha, just a gas exchange system measuring respiration and photosynthesis. So again, same graph on the left, and then on the right, now we're adding photosynthesis as they dry out. So the y-axis is photosynthesis, the x-axis is their moisture level. And you can see that photosynthesis for all of those mosses peaks at around 65 to 80%. So again, remember, 100% fully saturated, that CO2 isn't diffusing as readily into the leaf. So we can see that once it dries out a little bit, it peaks. The photosynthetic rate is much higher for the open habitat species on the bottom 2 graphs, and really high for Polytrichum juniperinum. However, kind of the main takeaway here is that these mosses are physiologically active over a wide range of moisture levels rather than the range of just their peak photosynthesis. I also measured light response curves, so measured their photosynthesis at different light levels. And just to kind of orient you, the bottom, the x-axis is light levels, 0 is no sunlight, about 1,000, 1,200 is about half full sunlight. And what's the craziest thing here to me is that yellow line, that's Polytrichum juniperinum. That is a really big photosynthetic rate. And that was really interesting to see. But we do see that the top 2 lines are the open habitats, and they have open habitat species, and they have much higher photosynthetic rates than the understory species in the lighter green and the orange. So the objectives of my warming experiment, I wanted to compare open habitat to understory moss species to see if they differ morphologically and physiologically to warming. I also wanted to look at how warming might affect the survivorship and growth of the mosses as well. My hypotheses for how these mosses might respond to climate change. We might see that open habitat mosses may be more tolerant of warming and precipitation because their habitat kind of predisposes them to that stress already. And with warming, mosses may be able to photosynthesize more in cold weather, which might increase their growth under elevated temperatures. But conversely, under warm weather, warming could lead to greater respiration, which is just carbon loss, leading to less growth. Warming could also shorten the period of time when mosses are at an optimum water content to photosynthesize. And open habitat species will maintain physiologically suitable water contents for a longer period of time than understory species. So this is my heating experiment setup. So the top, this graph up here, the heated graph, those are infrared heaters over mesocosms. So those are the 4 species in little tubs that I just collected in the field and put in native substrate. So on the right is the Polytrichum juniperinum and ceratodon, which are open habitat species getting full sun. And then under the shade cloth are the little understory species. And so I compared the heated, about 3 to 4 degrees Celsius, to above ambient temperature compared to the ambient. So no warming at all, just same setup, but no heaters. And I measured temperature with a weather station and a data logger. And then there's the entire setup, kind of a longer view of it. So they're kind of right next to each other in a greenhouse courtyard. So this is what they looked like in the tubs when I first put them out there. So the 2 understory habitat species, and then Polytrichum and ceratodon on the right. This is pretty interesting. This was in winter when it snowed. So this is the same day, same time. So this is the heated open habitat species, and then the ambient, just a few feet away, and they have a full snow cover on them. And this is interesting because we don't know really what this effect has on the mosses, reduced snow cover. We didn't really look into that, but it could be really important to look at. So I measured growth as the increase in area of each moss sample using an ImageJ program from NIH. And then this basically just allows me to adjust the growth for any variation in size among the relocates at the start of the study. So these pictures, this is the same moss sample in the same tub, pre-treatment on the left and post-treatment on the right. And these are both warmed species. These were under the heaters. But I basically just traced them in the program, and it measures how many pixels the traced shape takes up. So this is just post-treatment. So I'm going to show heated on the left and then ambient on the right, and then the survivorship in the middle and the species on the bottom. So ceratodon, you can kind of see both tubs look very similar, and every single sample survived, which is a win. Second one, we got another 100% survivorship. This is Theridium delicatulum. You can see both tubs, they grew a lot. I mean, each of these is 5 clumps, but you can't even really tell where they are anymore because they just sort of meshed together. Again, hypnum imponens, 100% survivability, and heated on the left, ambient on the right. And this one's interesting. This is Polytrichum juniperinum. Survivorship was 88%, which is still good, but it was the only one that didn't grow after the treatments. And you can really see how stressed it is on the left under the heaters. And basically, this kind of tells us mosses may be able to adapt to warming in terms of survivorship, but Polytrichum juniperinum may be more sensitive to these changes. So I'm still analyzing those numbers as well as photosynthetic measurements that I did. But I want to know, did warming increase the growth more than the non-heated treatment? Because they both did, all species did grow except for Polytrichum juniperinum, but I don't know if they grew more under warmed or non-warmed. So mosses in this region may have the capability of tolerating moderate warming, but long-term survival may depend on effects resulting from altered competitive interactions that can only be obtained by more intensive long-term studies in the field. And warming in high light combined may be more stressful than warming in the shade. And responses may be both species and habitat dependent, again, kind of requiring more intensive studies. And I'd like to thank all of these people, and especially North Carolina Native Plant Society, for helping me with this. And it's been really fun. And any questions? Yes.
With the severe rainfall and flooding that comes with global warming, do you
Yes.
think mosses would survive?
That's a good question. Yeah, so the question was about changes in precipitation with climate change and kind of predicting how mosses may respond to that. Yeah, so with changes in precipitation due to climate change, it may be more kind of severe rainfall and then more periods of drought kind of interspaced between. And I was initially going to look at that, but I'm only doing a master's project and that would have taken forever. But that I wanted to look at that really. But I don't know. I think with all of that moisture all at once, it could kind of, it may wash away nutrients more quickly, and then they have a longer period of drought in between, so that optimum time for photosynthesis is going to be shorter, and it's only going to be after that rainfall. So I would expect them to not do as well. It kind
I
of
guess I was
depends.
wondering if they go dormant during severe weather periods.
Yeah, they definitely do go dormant. Especially during drought, really. And then I'm not sure what they do under snow cover, things like that, or if they're fully submerged underwater, I'm not sure. But definitely with drought, they do go dormant, so I'm not sure. That'd be good to look at. Yes.
So from what I've understood from mosses, and I don't know a whole lot about them, but I've heard that they're hard to transplant and they're hard to get to establish. Did you have much problem with, like, I mean, you're measuring them drying out and taking them from their natural habitat and then uprooting them. Are you able to do those same sort of measurements you took out in the field to see if maybe just transplanting them had a big impact on them drying out?
Yeah, no, that is a good question. So the only one that I really had trouble with transplanting was Polytrichum juniperinum. And we kind of see that because I think it had a lot of stress when I uprooted it out of its natural conditions, put them in a mesocosm. And then most of them kind of did poorly. They survived, but didn't do as well. And that could have been the relocation stress. But the other mosses were very easy to relocate. I just copied exactly what the shade level was in the forest. I kind of took my light meter out and then put the shade cloth and did exactly that. I took their exact growing medium. So basically they didn't even know, hopefully, that they were being but then, so I did a process called dehardening, which is like a 2-week process of just going out to these mosses in the mesocosms every day and watering them and kind of getting them established and really spoiled before the experiment starts. So we did do some field experiments as well, but nothing with the warming. That's what we did.
So was there any way of like kind of tracking when the ones that did really well in the heated environment as far as recovery? When it wasn't in the heated? I'm just kind of thinking about the length of the, the heat and the
Oh, so,
non-optimum condition.
so tracking it's, so what do you mean?
I mean, just kind of ended there, or have you watched to see if it's back in its native environment that it has recovered?
Oh,
The time that it takes for it to recover.
after being heated?
Yeah.
Yeah, that's a good question. So, someone's asking about recovery of that Polytrichum juniperinum species. So I do still have the tubs and they're not being warmed anymore. We still have them. I haven't put them back out in their native conditions yet, but I don't know. I haven't looked at them in a while. So far, they're not looking great still. I think I don't know if they're going to bounce back. But yeah, that's a good question. I don't know much about moss recovery. But does somebody else have a question? Sure.
How much heating was actually provided? Was it similar to summer weather or more extreme than our summer weather?
Yeah, so someone's asking about how much warming was provided. So I did 3 to 4 degrees Celsius above ambient temperatures, and I measured that. So basically the heaters, whatever temperature it was outside, they just raised it that much. So the summers were hotter. So if we had a long streak of really hot week with no drought, those mosses under the heaters had that plus more temperature. Hopefully that answers.
Okay. If there's not any more questions, we'll go ahead and switch over to Erin. Erin,
Okay, awesome. Great. Well, that last talk was totally fascinating. I really enjoyed it. Go ahead and get you all started on a totally different realm. My name is Erin. I'm a PhD candidate at NC State and a 2023 Shinn grant recipient. And I'm going to be talking today about just a small part of my work, which the Shinn grant funded, recruitment of threatened smooth purple coneflower seedlings. And rather than taking y'all through one experiment from beginning to end, what I really wanted to bring you into was sort of the best part of science, in my opinion, which is when you're able to ask one question and have it lead into another question and lead into another question. And it's through funding sources like the NC Native Plant Society's Shinn grant that allow you to actually chase that rabbit. So for the sake of time, I'm going to skip my typical spiel about how amazing the Piedmont prairie ecosystem is. But this is the context within which all of my dissertation work happens. This highlight grassland system, which we think was once present throughout the southeastern Piedmont geophysical region. It is super diverse in flowering plants. There's awesome pollinator communities, and it's really hard to take a photo of it that doesn't include a power line running through it, because these days it's pretty restricted to rights of ways and roadsides and things like that. But here's a collage of some of my favorite photos of what I've seen while I've gotten to work in this system. One of the most charismatic plants that's present in this system is Echinacea laevigata, the smooth purple coneflower. It's now extant in just 4 southeastern states. Those are the Carolinas, Georgia, and Virginia. And we think that's largely due to habitat loss of this grassland system that it seems to be affiliated with. It's a long-lived species, which is self-incompatible, meaning that it requires insect pollination. So insect vectors to move pollen from plants which are distantly enough related to reproduce. And we think it's affiliated with semi-open to open grassland habitats. So a little more about those light conditions will come up later. So the listing history for this species begins in 1992 when it was formally listed as endangered. And then we had a species recovery plan come out in 1995, which has directed conservation efforts since then. There was an unsuccessful proposal to downlist to threatened in 2011. Those benchmarks for downlisting had not yet been met. But in 2021, another proposal to downlist came through, and this was taken up for having met the conservation benchmark set out in the recovery plan. And that brings us to today, where Echinacea laevigata is now listed as threatened. So how we got to this point was reaching a benchmark for downlisting, which called for 12 protected and self-sustaining populations across the range of the species. Really amazing work has gone into making that happen through various conservation partnerships, particularly our own North Carolina Botanic Garden has been really critical for establishing and augmenting populations with nursery grown plugs. This has been really great for getting those population counts up, but it's also kind of a case of helicopter parenting. So this is really labor intensive work, requires a lot of financial input, and it's not necessarily leading to these self-sustaining populations that Fish and Wildlife really wants to see in order to completely delist the species, which is the final goal for conservation of Echinacea laevigata. And having this non-helicopter parenting strategy, having these self-sustaining populations is going to be really important, not just for persistence of the species, but for resilience through time. So we need these self-replicating populations in order for the species to continue. As older plants senesce, they need to replace themselves. But it's also this sexual reproduction which allows for the release of genes for selection to act on. So we're in really turbulent times, environmentally speaking, right now, with ongoing climate change threats and also increasing urbanization in the Piedmont of the Southeast, which puts stress on these populations. And in order for them to be resilient through a longer time span, they need to be having this reproduction going on. And so we need to be able to measure population trends in this rare species. How I do this in my work is through population demography, which works a lot like a human census. So we go through populations, we count plants, we measure them, we tag them so that we can return to them in future years and monitor them. Monitoring these same plants through times allows us to determine whether things are dying, who's growing and maturing into reproductive adults, how much recruitment is happening, and reproductive rates, and such as how frequently plants are reflowering. And with all of that data, we'll be able to make estimations about how our populations are growing and thriving, or whether they're just sort of surviving at steady states of individuals which are already mature, remaining mature. I had another question that I wanted to bring into this study, which was about those variable light environments, which I mentioned earlier. So we find populations of Echinacea laevigata growing in really high light situations, and we think of that as being like optimal habitat. But we also have these kind of relict populations which are persisting in lower light habitats, like you can see on the right here. And there is a movement in land management to try to bring about these higher light conditions because we have these ideas about that's what the species needs to survive. But those management actions can, again, be costly, labor intensive, and also potentially harmful to the plants themselves and other wildlife. If you're bringing in heavy machinery for mechanical thinning, or you're bringing in herbicides to treat resprouts of trees, that could potentially have adverse effects for the ecosystem. So part of what I wanted to bring to this study was finding out whether that canopy closure environmental variable is having an effect on our demographic rates. Maybe our populations that are growing in the shade are still reproducing at comparable levels to their counterparts in lighter environments. So what that means for our field work is that we go through every year and we search for new plants in these demographic transects. We mark them sometimes with colorful nails like in this photo. We measure those new plants as well as old plants. And then at the end of the growing season, right before leaf fall, I go out and collect canopy closure measurements here using a hemispherical camera lens in order to sort of capture a snapshot of what the surrounding canopy is like. And we do this every year. So by the end of our study, we'll have these annual transitions of these known plants, and we'll have all of that population data that I talked about earlier, along with this environmental variable. So we'll be able to model our population trends, starting with our sort of baseline most important thing, the size of the individual plants in the experiment. So you can't ask a plant how old it is, and working in natural populations, we don't have an idea of when our mature plants first germinated, but size tends to be a really good predictor for some of our most important vital rates of plants in these populations. So we'll be able to model how dependent the size of a plant in, say, its second year of life is per its size in its first year. We'll be able to say, you know, how much does the size of plants in year one, how many flowering stalks they created, how does that influence the number of seedlings we have in year two? And then also included in those models, we'll have this environmental variable of canopy closure, which will test to see whether it's actually improving our understanding of those vital rates. So that's as much as I'm going to talk about the statistics that go into this project. But it all works out to be pretty elaborate. And we don't have all that data in hand. This study is going to go on for a few more years. And in the meantime, I want to talk about just some of the general trends that we've noticed in the seedlings that we're seeing when we're out there. The good news is that there are seedlings. That was a little bit in question because previously, when people had gone out in less systematic ways and looked for natural seedlings in these populations, a lot of the time they turned up empty-handed. But the good news is that in some of our populations, we're finding tons of seedlings. And these seedlings are surviving. We have about 2/3 of seedlings surviving their first year of life, which is honestly amazing. These are teeny tiny plants that manage to overwinter somehow. We also tend to find seedlings that are clustered together in these populations. So the best way when you're out surveying these transects to find a seedling is to first find a different seedling and then look around it for more. That seems to indicate that there's something really important about the microhabitats that seeds are falling into that's related to who germinates and grows into a living plant. And one of the things we think is most important is that we see seedlings which are occurring on bare earth. And I'll circle back around to that in a little later. We had something really awesome happen in 2023. So on the right here is an image of one of our sites called Williams. And for the first 2 years of our survey in 2021 and 2022, we never found any seedlings. There's over 100 mature reproductive plants at this site. Every year they create a ton of heads, which seem to drop a ton of seeds, but we were never able to find any seedlings. And that wasn't super mysterious because you can see that the canopy is really closed here. Those deciduous trees are dropping a lot of leaves onto the ground. So we didn't think of it as being optimal habitat for seedlings to occur in. And then in late spring 2023, we had a prescribed burn which was run through the site. We were actually a little bit worried about it because it was kind of late in the season for it, and we were worried it would just crush the reproductive effort of plants for 2023. But what we actually found was that the plants didn't mind, and there were 411 seedlings which turned up in our survey, which is obviously a massive improvement from zilch and zilch in the 2 previous years. And also at this site, now we have nearly 3 times as many seedlings as we do adults in our survey. So that just totally blew our minds. For a site which we had sort of consistently written off year after year, we were like, whoa, this is, you know, we need to figure out what went right, and we need to start making sure that that's happening at other sites. One of the cool things about Williams was that it had been a really small natural population of Echinacea laevigata, and there was a big augmentation at the site. So this was potentially a success story where, you know, there was this starting boost from our conservation partners that got the site off the ground, and it looks like it might take off running on its own, which would be amazing. So what was going on at Williams that was so great? We really wanted to dig into this idea of bare earth. We think of bare earth for many plant species as being really, really important. As you can see in this image on the left here, there's kind of this bare earth nursery, which looks like such a compelling environment. There's no competition from these big adult plants around these seedlings. There's not a lot of dead plant material, which is interfering with them. There's just this beautiful mineral soil that's available. Lots of light, and after a burn has come through, we expect there'll be lots of nutrients available for these seedlings to get a head start on life. So we had in our minds that this was going to be really important for why, after a burn at Williams, there was such great success. So we decided to dig into this litter question in 2 different ways in 2023. The first was through running a common garden germination experiment, and the second was through conducting field surveys for litter depths. I'll talk about this common garden experiment first. You can see on the left here, on the left of the left image, we've got a tray full of 6-packs, 2 trays full of 6-packs, one which is covered in a layer of leaf litter. Underneath that structure on the right, which is sort of this shade enclosure, there is 2 trays just like those on the left. And so what we have here is a factorial experiment to test the effects of shade and leaf litter on the germination of Echinacea laevigata. And we were expecting to see depressed germination in our shade treatments and depressed germination in our litter treatments, and maybe some interaction between those factors. This figure is courtesy of an undergraduate named Chloe, who I'm mentoring at NC State. And if you look on the left here, these two blue bars are our light treatments, and our red bars on the right are our shade manipulated treatments. And then our hashed bars have the litter added to them. And both of our litter treatments had depressed germination, but both of our shade groups did way better than either of the sun groups. And that was, so confounding for us. We weren't expecting to see that at all. And of course, everything is depressed from under that 90% germination in the growth chamber benchmark. That's just to get an idea of the average seed viability in the population to begin with. So nobody does as well growing in a growth chamber. Nobody does as well growing on actual soil than they do in a growth chamber. But still, those shade groups did so much better than we were expecting. Our first thought about this was that we did plant out this experiment in June, which is getting into the really hot, heat of the summer. Potentially those seeds were just getting totally fried in the sun treatments. And that's not really a naturalistic proxy for what we think is going on in the field. So we're planning on redoing this experiment in 2024 along a more spring germination timeline. So we'll be getting that started in March and April. And in the meantime, we thought as long as we're sort of recalibrating this experiment to be more in line seasonally with the light expectations for germination in the spring, we also want to take a closer look at making sure that our litter measurements are naturalistic. So we went through in 2023 and took these field surveys for litter depths, where we had this kind of blue domino pattern of locations where we sampled litter at every meter on our transect where we found seedlings. And where we found a seedling near one of those litter dips, we noted it. And then took additional depth measurements where we found seedlings that weren't near those domino points that we were sampling. With that data, we're able to say, what is the average litter depth along our transects, potentially suitable habitat for seedlings, versus where do we actually see seedlings? So for example, in just one population, these aren't huge differences in numbers, but they are differences that are present. We found an average depth of 0.5 centimeter of litter, but we were finding seedlings in only 1/5 of a centimeter of litter. So we're looking closely for those kinds of differences to see how much that microhabitat might influence seedling germination. And then we're going to take that leaf litter data and use it in our 2024 redo of this previous experiment. So the next question we had about what was going on with Williams was the actual fire itself. So this heat coming through. We know that Echinacea laevigata is really resilient to fire. Mature plants have this awesome thick rootstock that allows them to sprout back year after year and really soon after a fire. This image that I have here is actually the day after a prescribed burn. I don't really understand how this happened, but those plants were not bothered by the fire at all. They were doing just fine. But those were also really big mature plants. We don't really know how fire influences seeds and seedlings. So we really want to understand what temperatures can Echinacea seeds tolerate, whether there's actually a positive scarifying effect of heat. Maybe this is one of the ways that seed germination can be increased if we have those seed coats weakened. We're also really curious about the timing of burns and how that might affect population management. So for all of those seedlings that we observed in 2023 in Williams, we expected that those were the product of seeds dropped in fall 2022. Meaning that the seeds had managed to overwinter and then survive the fire in the spring. But with this experiment, what we want to do is test a few different scenarios. So I'll describe what we're going to be doing in the lab and then try to relate it to what that would actually translate to in real life. So the first scenario is where a population isn't burned. We simulate winter through this cold stratification and then expose the seeds to a mock spring with lots of light and warm temperatures, and then we'll measure germination. So that is what we'd expect to be happening in a population which doesn't burn in a year. And that's also the technique that's used when people grow up Echinacea at botanic gardens or in nurseries for outplanting. So we're also going to test what a spring burn would look like. And this is sort of the most common management technique for Echinacea laevigata populations. This is what we saw at Williams in 2023, was a cold stratification period simulating the winter, and then this heat in the late spring before exposure to spring germination conditions. So every time I have this fire icon on here, we're actually testing 3 different levels of heat because we want to get at that question about the intensity of the burn. The third scenario we're testing is a fall burn. If you have that heat exposure before the long stratification period through the winter and then spring germination, do you see a difference in germination rate or in germination timing compared to those other treatments? And the final thing, which is kind of a shot in the dark, is whether germination is possible without cold stratification following heat treatment in the fall. So this is going to be one of the ways that we think more about whether there's a scarifying effect of fire that releases seeds from dormancy. This is also something we're a little bit concerned about, given how much more difficult it is for a seedling to overwinter rather than a seed. So all of this taken together, I'm really lucky to have had the opportunity to explore so many different ways that seedling dynamics might be shaped in the system. And I'm really excited to be moving towards a more comprehensive understanding of habitat management to get to this self-sustaining population benchmark for potential delisting of Echinacea laevigata eventually. I'm super grateful to have worked with undergraduate students on getting these germination projects off the ground. All of this has been done in conjunction with Mike Coons at the NC Botanic Garden and with support from the North Carolina Native Plant Society and other funding partners. So I'm based out of the Irwin Lab and the Krings Lab at NC State. My contact info is at the bottom of the slide here, and I'm happy to take questions.
Any questions here in the room? Got one?
Yeah. Hi, Erin. It was a great talk. So I'm curious, your experiment where you're comparing shade to no shade. I'm curious where you got your seeds from, and if you think it mattered if you collected seed from a shade habitat for your shade experiment and sun habitat for your sun.
Yeah, that's an awesome question. And it actually, it's a really funny one to bring up. I'll answer the first part of the question before I get into the irony of it. So the seed which was collected and used in that sun and shade litter, no litter experiment all comes from populations which are in full sun, so without very much canopy closure. Collected across a pretty large geographic area, relatively speaking, all within one population, but from a number of individuals that we don't expect to be related, but ones which have been in a full sun environment for, we would think, multiple generations. And that was largely logistically based. This was just the best population that can spare the most seeds. We don't want to be taking seeds from populations which are smaller because we do want to keep that seed bank going and make sure that those populations have the potential to recruit and reproduce. So we basically have one big population that we're able to pull from for experiments like this. But I do have a lot of questions about how recent light conditions may have these shaping plants' capacity to tolerate different levels of sun and shade. So I had a project earlier in my dissertation, which was eventually scratched due to logistical concerns, that had me hand crossing plants from sunny environments and shady environments, potentially germinating the seeds, growing them up, and crossing them again to outplant and try to get an idea of the potential for local adaptation to those different light conditions. So it's something that I'm interested in for sure, but logistically not something we were able to address in that experiment.
Well, I don't think we have any other questions here. We can go ahead. Thank you very much, Erin, for that presentation.
Great, thank you for having me.
And we will go ahead and switch over to Lydia. Should we be good to go?
All right. Hi, thank you guys for having me. I'm Lydia Cox. I'm an undergraduate student studying biology at Duke University. I received the Shinn grant last year, and I'm very excited to share my research with y'all. My project is using herbarium collections to disentangle the effects of climate change on flowering phenology of native carnivorous plants. And basically, the research question for this is looking at whether flowering has changed in these plants over time and if this is tied to climate change. And my advisors for this are Dr. Natalie Kerr and Dr. William Morris with the Duke Department of Biology. So the species that I'm looking at for this study are 8 species of carnivorous plants that are native to North Carolina. And the first up is the Venus flytrap. or Dionea muscipula, which is actually only native to North Carolina. And the other species are 7 species of pitcher plants in the genus Sarracenia, which are native to North Carolina as well. And besides being really unusual and interesting plants just because they're carnivorous, many of these plants also face conservation concerns. 2 of them have been listed as vulnerable, 1 as endangered, and 1 as critically endangered. The rest are officially unlisted, but the International Union for the Conservation of Nature notes that they need more information to really assess the actual state of these plants. And so these species are threatened in part because carnivorous plants are especially susceptible to climate change, with 1/4 of carnivorous plant species worldwide predicted to be at risk for extinction. And one of the main reasons for this is that populations of carnivorous plants are often really small and sparsely distributed, which means that it's really easy to have habitat and population fragmentation that can be exacerbated by climate change. And additionally, many of these species live in primarily wetland habitats, which are extra threatened by climate change, by either sea level change or drought. And finally, the specialized ecologies of carnivorous plants that make them carnivorous make them particularly sensitive to environmental changes, and they are often the first species to disappear from ecosystems. And taken all together, these effects mean that carnivorous plants are very likely to have already experienced and continue experiencing impacts from climate change. So next, in general, climate change has effects on plant life cycles. Many aspects of plant phenology, or the timing of life cycle history events, are seasonal, and they depend on ambient environmental factors. So these processes are especially vulnerable to climate change. For example, phenology events such as the timing of sprouting, flowering, or fruiting in plants can change in response to climate variables like temperature or precipitation. And these changes in phenology can have cascading effects for plants and their life cycles. The different timing of important life cycle events may allow plants to expand their range and move into new territories, or on the other hand, it could force them to have a contraction of their territories. Additionally, in particular with changes to flowering, there's the potential to have a mismatch with pollinator species. And this occurs when the timing of plant flowering and the pollinators that they're required to pollinate them for them to reproduce get out of sync. And this can have really negative effects overall for plant life cycles. So to assess these impacts overall on carnivorous plants, I am doing a longitudinal study using long-term data from herbariums. And herbariums, if you're not familiar, are pretty much libraries or museums for plants. They store plants, usually in a dried form, along with information about the particular plant specimen, such as when, where, and how they were collected, what species, and other information like that for the use of future science. And CERNEC, pictured here, is the Southeast Regional Network of Expertise and Collections. And this is a consortium of 233 digitized herbariums. And there's a ton of information available here online. And with some refining parameters, like limiting my data to only specimens collected from North Carolina, only specimens collected since 1880, and excluding any plants that were cultivated in greenhouses or otherwise grown outside of their natural habitat. For the Venus flytrap and the pitcher plant species that I'm studying, there's about 2,000 specimens to work with for this data. And here's just a quick look of sort of in a little bit more detail what I'm talking about here. This is an actual herbarium specimen from CERNEC. It's a photo of a pitcher plant that was preserved at the UNC Herbarium. And sort of what's most important here for me and for this research is the specimen label, which tells a lot of information about the plant. And there's also, of course, the preserved plant specimen itself, which is another crucial part of the data. And there's also a scale ruler and a color control card, which are included in each specimen photo to really help with calibration. So a lot of the data specifics for these specimens can be taken from the specimen label or the specimen's entry on the website. But for some of the data, I actually need to access it from the photos. In particular, assessing whether the plants are actually flowering. That's the main focus of my research, is looking at flowering phenology change. And in particular, beyond assessing merely if the plants are flowering, I'm looking at flower stalk height, flower size, and trying to assess as well if the plants are healthy. So looking at if the whole plant was collected and measuring how large the leaves are. And to do this, I'm also using ImageJ software, which is free and developed by the National Institutes of Health. It allows me to calibrate the size of the scale ruler that's included in the herbarium specimen photo to actually measure in centimeters how large these different parts of the flower are. And this is super helpful, and it gives really accurate measurements. And yeah, it's really great for the data collection process. So here's a quick example of the data that I've been collecting from that previous specimen that I showed. You can see that I've got identification information, the species, location and habitat information, when it was collected, and different metrics about the size and quality of flowering and the size and quality of the plant as well. And thus far, I'm still in the process of data collection, but it's going pretty well, and I've been able to do some exploratory data analysis so far on the collected data, which I will show here. So first, these are some maps showing the distribution of counties present in the herbarium specimens. So basically, any county on these maps that is in green, there are specimens for these species in the herbarium database. So for these three species, the Venus flytrap and Sarracenia flava and Sarracenia purpurea, they have the largest representation or the widest distribution of data that is present on the herbarium website. And moving on, Sarracenia rubra and Sarracenia minor, these species are sort of mid-range as far as distribution in the herbarium database, but they still span quite a few counties that there is data available from. And finally, for Sarracenia jonesii, leucophylla, and oreophylla, these have a lot fewer counties that are represented in the data, some as few as one. And overall, there's a couple reasons why there's sort of these discrepancies in the data. Fundamentally, some of the species just have a really contracted range compared to others, so there's just less area to draw on where they can be collected. Additionally, it's sort of just a bias that is present when you're working with a historical data set.
Hey Lydia, we lost your audio there for about 10 seconds. Can you go back to right after you said the county size is contracted?
Yes. So yeah, some of the specimens are only present in some areas because they don't have a very large range. And sometimes it's also due to the bias that's inherent in a historical data set. Because they may be, back in the 1800s, there just wasn't data collected from these particular counties. So it's partially due to the natural range of the organisms and partially due to the collections that are available. And next, I have a chart of the number of herbarium specimens for these species that were collected each year since 1880. And there's kind of a lot going on with this chart, but there's a couple important points that stand out. First, you can see there's a lot of variation between years and how many specimens were collected. In general, there's sort of a bell curve, but you can see there's some years where no specimens for any of these species were collected. And there's some years, particularly in the 1970s, when there were a lot of specimens collected. So this is, again, sort of just inherent with this type of data set. Some years there's a lot better data than others. And you may also notice that there's an uneven spread of color. According to the legend here, each species has a different color, but in this graph, some colors are represented a lot more than others. this shows another aspect of the data that I've collected, that there are simply just more records for some species than others. And this is, again, some species are a lot more rare and less likely to be collected than others, and partly some were just happen to be collected more often. But overall, there's fairly robust data, even dating back to the late 1800s, which is great for looking at flowering in the very long term. And going forward, I plan to do more in-depth statistical analyses for these data. In brief, I will use R statistical coding to calculate the long-term average of flowering time, as well as deviations from this average. And I will also build models to predict flowering phenology shifts for these species. And using these models to assess flowering quality, this can be linked to long-term climate data that is available from climate databases. And ideally, this type of analysis should reveal some very helpful information. In particular, it should show whether the time of year that these plants flower has changed over time, and if the quality of that flowering has changed. And additionally, if there's any correlation between this change and climate change, as well as the strength of this effects. And as I mentioned at the beginning, these plants do show conservation concern and are likely to be impacted by climate change. So if we can quantify or at least identify a relationship between climate change and changes to phenology that may potentially affect range or pollinator relationships, it could be very helpful for conservation efforts going forward for carnivorous plants. And finally, I'd like to thank the North Carolina Native Plant Society and the Shinn Grant for making this research possible with their generous support, and to acknowledge the role of CERNEC and the Duke Department of Biology in facilitating this research. And these are my references. And thank you again, and if anyone has any questions.
I do have a bit of a question. I know you said, so most of the work that you're doing is based off of herbarium species. Are you also doing it based off of many live locations, or is it mostly herbarium species, herbarium samples?
Yeah, as of now, just limiting it to herbarium specimens. There are some that have been collected up into the 2020s, but it's definitely more of looking at sort of a historical data set and not field work.
Well, thank you very much for that presentation, Lydia. And we will go ahead and jump over to Lauren.
So, hello everyone, my name is Lauren Wilhite, and this is my thesis work, Long-Term Survivorship and Species Invasion on a Restored Urban Stream. So for a little bit of background, urban stream restoration is increasingly being used in mitigation, but post-restoration monitoring is minimal, or and even fewer are done like on a long-term basis. In North Carolina, there's a 5-year requirement that the Division of Mitigation Services requires, but you can imagine that that 5-year mark isn't really representative of a full like plant community assessment. So in these short-term monitoring, they usually find that fast-growing species are like dominant, which makes sense if you're only looking at a 5-year scale. And then often in restoration work, there's this hypothesis, the empty niche hypothesis. And that's because they're looking to see how they can kind of plant to reduce the amount of invasion occurring. So the idea is just to plant species with similar functional traits to invasive species so that the niche is filled and that the invasive species can't come in, hopefully. So my study site was Rocky Branch. This was a stream restoration project done on NC State's campus, and it used to be one of the most polluted streams in the state. Before it was the most polluted stream in the state, it was just kind of a pile of construction dirt, so you can understand that that's a pretty disturbed site. It was restored in 3 different phases over time, so it gives a pretty unique opportunity to look at a more long-term plant community development rather than that 5-year mark. And then we have access to planting lists for each of these phases, so we can kind of compare what was initially planted with what's now seen in that phase. So here is the map of those 3 phases. Phase 1 was finished in 2002, phase 3 was finished in 2006, and phase 2 was finished in 2006, and phase 3 was finished in 2010. So obviously this like 22 years in phase 1 is a lot longer of a timeline than that 5-year mark. So hopefully it'll give us some better information about what this plant community looks like now. So my question is, what is the vegetative success of Rocky Branch Restoration Project in terms of planted and invasive species? So you might ask, why is this important? And hopefully this will inform other stream restoration projects in similar environments, like in the southeastern Piedmont area, about what species are thriving in restoration, what species may be effective at preventing invasion by filling niche space. And then hopefully we can make planting lists accordingly for future projects so that we don't waste time, money, and resources on things that don't work or things that aren't going to be that effective. And then hopefully we can provide some knowledge about what invasive species should be heavily targeted for removal based on how much impact they have in riparian zones. And then we can better understand the ecological consequences of not carefully managing invasive species. And there has been some management done to this site, but it's more of like short-term efforts on like a few species rather than continuous management. So we can kind of see what are the consequences of not having that continuous management present. So my methods, I had 10-meter wide transects that ran from tree line to tree line. The length varied depending on where I was, because the floodplain could be wider. Certain phases are smaller than others, so that depends. And then within each transect, we counted the number of stems at breast height for all the species present. And then we categorized them based on if they were planted, volunteer native, and volunteer non-native, and invasive. So cover class was also collected based on tree, shrub, and herb layer, and also cover class for species by invasives. And when I say cover class, I just mean like the amount of cover it takes up of the transect in like terms of percentages. So you've got groups like 1 to 2%, 25 to 50%. So I just kind of assigned a percentage of how much that was taken out. And these transects, we did one transect and then skipped two, did one transect, skipped two, to try to get, a good distributional across the entire space. But for the in-between sections, we went and did presence-absence data on the planted species, just so we would have a full survivorship of the planted species. So I mentioned that the lengths vary. You can clearly see that here. Phase 1 and 2 were a lot longer in terms of the floodplain when compared to phase 3. So there was a little bit of relativizing that I had to do with this data. So some of the general findings. In terms of the stem distributions, the invasive layer kind of took up half of each of the phases, which is interesting to note because this is, 14 years versus 21 years, and they still have pretty similar distributional across invasive, volunteer, and planted, with volunteer and planted kind of each taking up a quarter of each of these phases, and then volunteer non-native kind of taking up just a sliver. So pretty interesting there. We also did indicator species analysis on each of these phases to kind of get a good idea of what each community was like. Phase 1 was a mid-successional community with more of a closed canopy that had a lot of bottomland species like pawpaw and redbud. They were a little bit drier in comparison to the species that we found in phase 2. Phase 2 was a mid-successional community as well, but it had more of an open community or open canopy. And it had a lot more floodplain species that were a lot wetter, like box elder, smooth alder, river birch, more of like wetter species in that phase. And then in phase 3, it's the youngest community, early successional stage, sort of still, with tulip poplar and American elm being the ones that popped up. And these are both light-seeded species that grow pretty quickly, so that makes sense for the earlier successional stage. So going into planted species, the survivorship is pretty high, considering that some of these species aren't really necessarily meant to survive. There's like some temporary seeds that aren't really meant to survive. So 62% is pretty good, 64% is pretty good. Phase 3 is a little bit on the downside with 40%. And I think that if we think back to like how big the floodplain was, it kind of makes sense it's not doing as well, since it just has so much disturbance and not enough floodplain to kind of mitigate that. And then it also is the only, it's like in the heart of campus where the other two phases aren't. So it has a lot more disturbance with people as well. So that can kind of contribute to its low survivorship. So these were the most common planted species in terms of the number of stems. These are wax myrtle, Carolina allspice, American elderberry, tulip poplar, and sugarberry. So we can see with these top 3, they were all multi-stemmed shrubs, grow pretty quickly. While tulip poplar and sugarberry aren't necessarily shrubs, they still are pretty quick growing. So we can see that that's pretty common amongst these top 5. And then additionally, in terms of the species, they also had high frequency across all those transects. So they had pretty high importance values as well. So here are some of the shared traits that I found amongst those successful species. They were all designated as facultative wetland species, which tends to be what's preferred in restoration projects because they can handle a little bit more like dryness and they're not obligates. Additionally, they were all had high amount of fruit and seed abundance, and they were all able to be propagated by bare root, which kind of indicates that they're a little bit more hardy species. And then I'm gonna, over the next month, kind of see if there's any more additional traits. So there might be more to come on that front. So these were the most common volunteer native species. Now, I underline laurel cherry because there's a lot of debate about that. I'm sure some of you might know that laurel cherry is sometimes considered to be not native. So I included the top 6 into this instead of the top 5 here. So I have laurel cherry, loblolly pine, black locust, American elm, sweet gum, and southern sugar maple. Now, most of these can be pretty much considered volunteer species, like normal ones that pop up a lot. Southern sugar maple, however, is a little bit more surprising than the rest and not one that I often saw in other restoration projects. So I thought it was pretty interesting to note that it performed so well at this site. Okay, so these species had the highest abundance in areas with the lowest amount of invasive cover. So in this case, low invasive cover was just less than 25%. So this includes black haw, northern catalpa, withrod, American elm, hawthorn, Shumard oak, river birch, and black willow. And I think an interesting thing to ask here is whether these species are more vulnerable to invasive species, and that's why they're not in the same plots with a lot of invasive cover, or if, going back to that empty niche hypothesis, they are filling up that niche space and are preventing the invasive species from occurring. This wasn't something I could necessarily look into yet, but something for the future to kind of keep considering, and maybe future research can be done on that. So going into the invasive species, looking at overall cover across the entire phases, we can see that there's pretty high cover. Phase 1 covered 57% with invasives. Phase 2 had 66% invasive cover. Phase 3, 39%. So phase 3 is a little bit lower, but still decently high. We also did indicator invasive species analysis to kind of see which invasive species were more for each successional stage. So phase 1, the mid-successional closed canopy area, had English ivy, Amur honeysuckle, Chinese privet, and white mulberry, which these are more like shade-tolerant species, which makes sense for the closed canopy area. For the phase 2 mid-successional open canopy, there's a little bit more of a mix between shade-tolerant and shade-intolerant species. And I think that's just because there's obviously more canopy gaps, but there's still a lot of shade in the area because there's taller species present. So we've got like porcelain berry, leatherleaf mahonia, thorny olive, lespedeza, creeping lily turf, Callery pear, and Chinese wisteria. So more of a mix there. Here we have phase 3. It was the early successional stage, and the only species that popped out was Japanese stiltgrass. And that makes sense just because we were talking about the small floodplain. There's a lot of soil disturbance that happens, and that is prime ground for Japanese stiltgrass. So these were the highest impact species by cover across the entire project. English ivy, glossy privet, Japanese stiltgrass, Callery pear, and Chinese privet. To me, this is not a very surprising lineup. They all rank as #1 significant threat in North Carolina, and they're pretty common from what I've seen in other restoration projects. So these are definitely some high impact species for restoration. Going a little bit into a deeper dive on each species, we've got English ivy. This seemed to be a biggest problem in phase 1 and 2. We can see that with the 21% of it covering phase 1, 17% of it covering phase 2. Pretty high when you're considering it's just one species covering 20%, 21% of an area. And this seemed to be more of a mid-successional concern since 1 and 2 were the mid-succession areas. So it had some good shade tolerance, and it was overall a pretty high driver for cover in invasives. Glossy privet was another big issue, and this was an issue across all phases. We can see with the percentages on this one, they're all pretty similar, 15, 12, 10. There's not really like a standout, but phase 2 was pretty high. So this seemed to be a problem across all successional stages, was a high driver for cover overall. And I think a main reason why it was such an issue was because it was planted on the surrounding campus, which provided a lot of seed source and allowed for the invasion to occur. Japanese stiltgrass was another big issue. Phase 3, as we mentioned, an early successional concern. And that's just because like the soil disturbance kind of stops in those later successional stages when those bigger trees kind of have more root and have more soil stabilization. So you can see that they still have a pretty high cover here, but they also have some presence in the other 2 phases, and that's mostly was in just canopy gaps. So yeah, more of an early successional concern. Colliery pear was another big issue. That was particularly in phase 2. We can see that this has more of a difference with 8% being pretty high. And this was like an open canopy problem. And that's because they pretty much became the canopy in this area when they could. So the species was pretty just large canopy trees that kind of just took up a lot of cover. And this was also planted on surrounding campus, which is why it was such a big problem. And then lastly, Chinese privet. So this was the biggest issue in phase 1. So it seems like it's more of a later successional issue when the canopy starts to close a little bit more because it has that high shade tolerance. And we can see that it has its biggest impacts in 1 and 2. So shade tolerance. So what are the big takeaways here? So here's the overall recommendations for restoration based on this project. So first and foremost, consider your environment and adjust your planting plans accordingly. You want to make sure there was a couple of species they planted that were just clearly not going to survive, were much too dry. So obviously those are like, okay, make sure that you're planting based on where you are. Additionally, plant species that can quickly kind of take up space. Make sure you diversify those species, but species like that have shrubs with multiple stems or high abundance of fruit and seed and hardiness, like with that propagation of bare root. So those traits that from those most successful species we saw. And another thing, consider other projects like kind of in your vicinity and take note of what maybe their unexpected volunteer species were, because those could be pretty successful in your project as well. If you decided to plant them, they could take off. For more slow growing planted species, make sure that you allocate like additional management. As you saw from this presentation, there really wasn't a lot of those slower growing species being dominant. And that's just because they didn't really have a lot of chance to outcompete those faster growing species. They just kind of took up all the space, and those slower growing species didn't stand much of a chance. So if you're going to plant those and you want them to thrive, make sure that you allocate accordingly. And then of course, I mentioned doing that further research on those species that were in the less invaded areas to see if they were preventing invasives or if they were maybe just vulnerable. And then targeting those 5 highest impact invasives that I mentioned. Make sure you allocate your resources to maintain any known populations. And it's crucial to manage those initially while the plant community is establishing. And then if it gets out of control, usually bottom-up removal is recommended, where you start with the herbaceous layer, move up to shrub and then tree. And then lastly, if resources permit, which usually they don't in restoration, you don't have a lot of leftover money, but if you do, there was those additional invasives I mentioned based on the successional stage that you can go ahead and target. So with that, I want to acknowledge my advisor, undergraduate assistant, and of course the Shinn grant for allowing me to do this. And are there any questions?
I've got a question. So when I know you're monitoring in each of these locations for how many invasives there are and kind of how well they're doing, is there any effort, or I guess like at what point would there be efforts to then go back in and remove those invasives, or is it going to have too much of an impact on the research that you would avoid that?
Yeah, so I think now that the research has kind of been done, we're starting to move in a direction where I make some specific recommendations to land management at NC State. Because when I've talked to them before, like I mentioned that they had done brief sort of management, they just kind of run into the trouble. They don't know where to start. They don't know what the highest impact invasives are. Like they just are kind of overwhelmed with the state of things. So this was kind of to serve as a way to be like, I've identified those things and here's what you can do. So we're definitely moving in the direction to start trying to do that. We're even starting to incorporate, there's a introduction to environmental sciences class at NC State that I TA for, and my advisor is the professor for, and we're going to do like a invasive removal day with all the students. And we're going to go ahead and target some English ivy because, as I recommended, bottom-up removal is the best way to kind of go about this. So we're going to start with that herbaceous English ivy and start ripping stuff up. So yeah.
Oh, you have two other questions?
Yeah, you might have mentioned this, but what was the percentage of invasive species there before the project started?
Yeah, so we don't have much data on how, like, before my kind of inventory, they didn't really have any data on like percentage. So currently we know that there was like a, let's see, I can just go back to that table. So now we know that phase 1 has 57%, phase 2, 66, phase 3, 39. But before me, there wasn't much data. All they really had was just they identified a couple species that seemed to be prominent, but they didn't really say how much. So unsure. But as of now, these are the percentages we know of.
But before the stream project began, do you know what the level of invasives might have been back before the project started? Like what's in the seed bank?
Oh, sorry, I think we lost your audio there, Lauren. You might be on mute.
Oh, okay. Can you hear me now?
Yep.
So before this project, there was actually like no, before the stream restoration project occurred, there was like nothing there. It was essentially, they filled it in with construction soil and it was just kind of like there. Oh, did I become big?
Yes, I the spotlight. Sorry about that.
No, you're fine. I was just like worried that I dropped out. So yeah, before that there was essentially no vegetation, and it's really interesting to look at because there are like some old photos of it, and it's just nothing. It's just like campus and buildings and stuff. So very amazing what they turned this into, even if it was pretty, you know, polluted. They eventually obviously cleaned it up. The water is great now. There's invasive species, but there weren't any before that. So it seems like most of it is just from surrounding seed source that they invaded from.
Okay, thank you.
Did you have a lot of poison ivy coming up?
Yeah, there was a decent amount. It was mostly, I feel like English ivy kind of was taking up all the space where the poison ivy was, or like could be, but there was definitely like a good amount.
And what did you do with the poison ivy since it's not really invasive, it's native? Do you leave it there or?
Yeah, I think the plan currently is just to kind of leave it alone. Yeah.
Okay.
Hi, could you explain why you're recommending bottom-up invasive removal at this point?
Yeah, so this is actually, I went to an invasive like plant council meeting, and there was a guy who worked with like, I think it was Wildlands Engineering, and they like specialize on restoration projects and heavily invaded ones at that. So he had recommended that in the area, usually it's good to do bottom up, because in his experience, whenever you started kind of with the shrub or tree layer, it opens up, you know, the canopy, and then those species like stilt grass can just like kind of take off. So if you don't address the native like herbaceous layer, or the invasive herbaceous layer first, usually clearing up space via like shrub and trees kind of just makes your herbaceous layer worse.
Well, thank you. I don't think we have any other questions here. I will go ahead. If you don't mind turning the lights on back there. There we go. Throw the camera on the ground. Okay. All right. Thank you all for joining us today. Thank you everybody who joined via Zoom, and to our presenters. The research you're doing is very exciting. It's really cool to hear just all the different sorts of questions people will ask when they look at our native plants. So it's really exciting to see all the different approaches to this.